Chapter 10's lecture. Please have your outline for Chapter 10 in front of you so that we can reference the same notes as we go through. The first part of this is basically who discovered DNA. So the molecule of DNA was not found out until in the 1950s, and there were two people responsible for the discovery. So I want you guys to remember Rosalind Franklin and James Watson with Francis Crick. So Rosalind Franklin was the first. She worked on DNA molecules. She took an X-ray image of DNA and she revealed that it was a helical structure. So that was one big step. And then James Watson and Francis Crick worked on exactly what the molecules were and how they fit together, and came up with the model of DNA that we know today, which is the double helix. So they got the Nobel Prize for discovering the molecule of DNA, but Rosalind Franklin was never mentioned, so we want to mention her, that she did the legwork for Watson and Crick to build on. So when we look at the letter B, Watson and Crick's model, this is everything that is in the structure of DNA that you guys want to know. So the first one is that it is a double-stranded molecule, so each side of the molecule contains one strand, and each strand has a 5' end and a 3' end. The 5' and 3' refers to the free carbon on the sugar. So let's review really quick, what is this unit of DNA? So what's our building block of DNA here? Remember the word nucleotide, right? So this is a nucleotide in here. There is a phosphate group. That is attached to a sugar. Remember, we call this a pentose sugar in class because it's 5 carbons. In DNA, in particular, this sugar is deoxyribose, right? So this is where DNA gets its name, the deoxyribonucleic acid. And then we have that attached to a nitrogenous base. In this case, they drew a thymine. And we use the word nitrogenous because you can see these nitrogens, right? There's an N here, there's another N here. So every single base has nitrogen groups in it. So if we look at how we are going to build on the sugar, what you need to understand is that the carbons are numbered. So the cursor where I am now, this is 1, the number 1 position. In organic chemistry, the carbons with the biggest group attached to it gets the first position, so number 1. So since the base is the biggest group here, this is our 1' carbon. Then it just goes around in a circle. This is 2', 3', 4', 5'. So when the ends of the DNA strand is called the 5' end or the 3' end, what that's referring to is the fact that when you attach one nucleotide to another one, you either attach it here at the phosphate. So let's take a look at this strand, right? We can see that there's a phosphate attached to our base, and the base here is attached to our second nucleotide. So the nucleotide has a phosphate attachment to that base. So it's the 5' end. The phosphate group is attached to the 5' end. So we just call this the 5' end. And then where here at this point, right, we can see the phosphate group is attached to the 3' end. So this carbon here, that's why there's a little line here. The 3' end is what attaches to the next nucleotide down. The little line here indicates that this phosphate group on the 5' carbon is where the next nucleotide attaches to above it. So if we look at this strand up here, right, the phosphate group is left. This is the last nucleotide on this strand. So there's no more nucleotides above it. So what's left hanging or free would be considered the 5' end. So this, what you're looking at here, is a 5' end because there's nothing else that's attaching to the phosphate group. The phosphate group really is sort of speaking for that 5' carbon. Now if we go to the other side, right, what is left open here? Well, not the 5', right, because the 5' is attached to the phosphate group, and the phosphate group has the nucleotide attached above it. So what's hanging and open is our 3' carbon, right, our 1, 2, 3. So this is considered the 3' end. So in DNA, the molecule of DNA, let me show you the other slide here. It's a little bit more detailed. If we look at one strand on this side, let's see if you guys can identify what end this is. What end is this nucleotide here? So we have a free phosphate group. There's nothing attached to the phosphate group, so this is our 5' end. And always on one strand, if there's a 5' end, then the other side is going to have to be the 3' end. All right, that's how it works. Now, if you go and look at the other strand doesn't look exactly the same. It's actually flipped upside down if you take a look at the way that the sugars maybe are oriented, right? This is upside down. So what we call this arrangement of the two strands is anti-parallel. That's actually number 4 in your outline. The two strands of DNA are anti-parallel. So let's see, what is our end here? So let's take a look at the nucleotide. Is this the 3' end or the 5' end? So if you answered it's the 3' end, you are correct because there's nothing building on this sugar group here. So this is the 3' end. And then on the opposite side, you know if that's the 3' end, then the other side is going to be the 5' end. So now, this is the sort of takeaway. You have a double-stranded DNA molecule. The two strands are going to run anti-parallel to each other, right? This one is kind of going, let's call this right side up. This one will be upside down. Also, on the end of DNA molecule, so if this, you're just looking at this end of the DNA molecule, if there's a 5' end on this side, the other strand is the 3' end. Let's go down here. This is the 5' end. We go on the other side, this is the 3' end. So because these strands are running in different directions, like a 2-lane highway, right? The cars are running in different directions, then you're always going to have one 3' end and one 5' end on one side, one 3' end and one 5' end on the other side. Okay, so that's what number 1 is talking about. So double-strand molecule, each strand has a 5' end and a 3' end. They refer to the free carbon on the deoxyribose sugar, and that's basically all you need to understand about that. Okay. Now, number 2 says that the two strands are complementary, right? So now you have to understand that the bases, which are facing each other in the center, are always going to pair bond in a very specific way. All right. This is number 3, the base pairing rule. If you have an adenine, right, an A, on one nucleotide, the nucleotide across from it will have to be a T, a thymine. So adenines always bind to a thymine, always. If the nucleotide happens to have a guanine as its nitrogenous base, the other base on the other side will be a cytosine. So C always binds to G. Always. And that way, you know, if you had one strand of DNA, you had A, G, T, and C on one side, then you know what the other side is going to be because they always have to pair bond in this way. So T, C, A, and G. You also need to know the dotted lines that hold those bases together are called hydrogen bonds. Okay, so that's a hydrogen bond. Hydrogen bonds, if you remember, they're not super strong. They can be broken, but they are definitely a force that keeps those two strands together. All right, so I think we're done with letter B. So we went through the double-stranded 1, 2, 3, and 4. We went through the hydrogen bonds. We're going to turn the page and look at DNA replication. All right, so let's look at, so this is a nice, oh, let me mention a couple other things about the DNA molecule that you want to know. So notice our rotating image of DNA, right? So this entire structure, the shape of it, you would call a double helix. And what we say is that the rungs, if this was a rope ladder, right, and you twisted the rope ladder, the ropes where you put your feet, right? The rungs, those are the nitrogenous bases. So they face each other and they hydrogen bond in the center. And then we call the backbone, right? The backbone of DNA. Let me go up to this one. So the backbone of DNA where you have the sides, we call those a phosphate sugar backbone. So phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar, those create the backbones, whereas the center will have your nitrogenous bases. All right. So here's your concept check. What is molecule A? All right, the answer is C, it is a deoxyribose. So remember our phosphate sugar backbone. The circle is a phosphate, there's a sugar. Phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. So these sugars in DNA is going to be deoxyribose, not ribose, right? Because we have a double-stranded molecule in this image. It's DNA. So let's identify the other features here before we move on. So B, right, those would be nitrogen spaces. C would be the phosphate group. And then D, which is kind of invisible here, there's nothing shown, but those would be hydrogen bonds, right? Hydrogen bonds in between our two bases. All right, let's look at DNA replication. So first of all, I need you to understand why DNA would replicate and what it means. So DNA replication means that the DNA is making a copy of itself. Okay, it's making a complete identical copy, just like a carbon copy or Xeroxing something, you're just making a copy. Why? Because in this image, cells reproduce themselves, right? So let's say this is one of your skin cells, right? You have a cut and you want to replace the dead or the missing skin cells with new skin cells. So this one cell here is going to have to reproduce. It's going to have to make more cells. So all the DNA that's in the nucleus of the cell is going to have to double. And then when it doubles, the entire cell is going to have to split in two. So at the end of the process, you're going to have two cells with the identical copies of the entire DNA genome in each cell, which is quite remarkable. Okay, so that's DNA replication. And let's talk about the semi-conservative replication.